first speaker for session one. Very pleased to introduce Hunt Potter, an old friend, director of the Florida Alzheimer's Disease Research Center, and is the Eric Pfeiffer Chair for Research on Alzheimer's Disease. Hunt is an iconoclast. He's thought about problems differently than many other people, and I think his contributions are quite significant, and we're delighted to have you, Hunt.
Thanks. Thank you for inviting me, and I also say that my memory is still good enough so that if anyone wants to ask a question in the middle of the talk, I'd be very glad to answer it and then get back on track after that, or we can leave the questions to the end as you like. The take-home message that I have for today is that instead of considering Alzheimer's disease and Down syndrome as two separate disorders, in fact, they're exactly the same disorder. And I will leave you with some data that we've collected over the last 20 years, not only in our lab, but in other labs, to indicate that. So to take to heart Bill's dictum that we should think about the Alzheimer's disease and Down syndrome as a series of gaps that need to be solved, I can say that our field of study is the role of chromosome segregation defects and microtubule defects in both Alzheimer's disease and Down syndrome. We have strong data. We have a new molecular drug target, which I'll tell you about. And the weaknesses, I think, are mostly in education and funding, which has been indicated already. There is insufficient funding for Alzheimer's disease, but there's vastly inefficient funding for Down syndrome. And one of the points that I would make is that if we have a treatment for Alzheimer's disease, we might treat somebody for 5 or 10 years at the end of their life. But if we have a treatment for Down syndrome, they're going to be treated for 60 years. So that even though there are 400,000 Down syndrome and say 5 million Alzheimer's patients in the United States, the ratio of importance becomes much more equal when you realize the length of time that we'll have to be treating a Down syndrome individual to bring up their status to normality. And directed drug development and clinical trials, I think, are some of the biggest gaps that we need to fill. Okay, so I'm going to just give one sort of background slide, which many of you already know about, which is that there is an amyloid cascade hypothesis for Alzheimer's disease, but it also, of course, refers to Down syndrome, which is that there is an amyloid precursor protein encoded on chromosome 21. Its cleavage by two proteases, beta and gamma secretase, generates the A-beta peptide, which is the start of a whole cascade of problems that occur in Alzheimer's disease and in late Down syndrome. The physical manifestation are the amyloid plaques, but the actual toxic agent may be a precursor to those plaques, such as an A-beta oligomer. We certainly need to know why this leads to the cognitive decline in Alzheimer's disease and late Down syndrome. And one of the things that is clear that pathologically, the two diseases are very similar. Here's an example of a 12-year-old, 17-year-old, and 29-year-old Down syndrome individual that Cindy Lemire reported some years ago, just to illustrate that the pathology in the brains of a Down syndrome individual can start very early in the disease, even if the cognitive deficits may not manifest in every individual and not into the 40s or 50s. In Alzheimer's disease also, the disease probably starts in the 20s and 30s and doesn't manifest until late age. So these are two problems that we need to look as early as possible in. The other thing that is true of both Down syndrome and Alzheimer's disease is that there is neuroinflammation in the brain. This can be detected in Down syndrome even in the fetus, where the number of IL-1 positive microglial cells by Sue Griffin's work is probably 30-fold higher than normal. That neuroinflammation begins a cascade that contributes to the cognitive deficits in Down syndrome at all ages, and also contributes to Alzheimer's disease in the sense that the activation of microglia and astrocytes around these plaques lead to the production of proteins that are necessary for the production of those amyloid deposits. Two of those proteins are apolipoprotein E, which we all know about, and anti-chymotrypsin, which some of us know about, which the genetic analysis of mice with and without these proteins being expressed show that if you don't have one of these and you still make the A-beta peptide, you essentially have no amyloid deposits and no cognitive deficits. So the A-beta peptide, which is sort of the boogeyman that we're all afraid of, in fact is totally harmless by itself. It needs neuroinflammation to generate these amyloid deposits shown here. Intermediate proteins are expressed with anti-chymotrypsin alone or ApoE alone are intermediate in the levels. And if you test these mice, this mouse is totally normal. And this mouse, of course, can't find platforms and other bases at all. So there's inflammation in the Down syndrome brain and the Alzheimer's brain, and there's overexpression of A-beta, and together they cause the late problems. Now, looking at these data, we came to a conclusion that Alzheimer's disease was really a form of Down syndrome as a hypothesis, which was that perhaps during the course of the life of an Alzheimer's patient, they developed trisomy 21 cells, and that those contributed to their Alzheimer's disease as they aged, and that's why Down syndrome also developed Alzheimer's disease. We published a theoretical paper, which upon its first submission received this one-line rejection. I wouldn't recommend publishing this manuscript anywhere at any time. As you'll see, we've spectacularly shown this reviewer to be non-prescient, but this is the problem that some new ideas face. Okay, so if we're going to count chromosomes in Alzheimer's disease and Down syndrome, we have two major techniques. The metaphase spread shown here, where you can just count chromosomes and you can tell which ones are 21 by their staining. Or we can do in situ hybridization, where we use a fluorescent dye, which is made of DNA and which goes and finds its cognate DNA on the chromosome and can count, in this case, say 2 chromosome 21s in this cell, which is, of course, normal. When we applied these techniques to, for instance, fibroblasts from Alzheimer's patients, we found that every class of Alzheimer's patient, both sporadic and genetic, had an excess of trisomy 21 fibroblasts. And that finding has been replicated by other labs in the brain and in the buccal cells and the lymphocytes of Alzheimer's patients. Some of the best data are Familial, shown here in lymphocytes, and by Thomas Arendt, shown here in brain cells. And essentially, the number of aneuploid cells in an Alzheimer's brain is about 30%, of which a third are trisomy 21. So Alzheimer's disease is a mosaic form of Down syndrome that is developed over the course of the life of the individual. And that means that if we study Down syndrome, we're studying Alzheimer's disease and vice versa, and that any drug that we developed in one situation may be applicable to the other. Yes?
So how many cells are aneuploid in a fraction of trisomy 21 aneuploid?
Okay, in the brain of an Alzheimer's patient, about 30% of neurons are aneuploid, and a third of those are trisomy 21. Yes?
What's the situation in normal subjects?
The normal subjects are usually around 5 or 10% aneuploid, possibly a bit less depending upon other things. And we don't know whether that's a true aneuploidy or whether that's it's a sort of background defect in the technique. Good question. Yes, sir. Cortex, hippocampus doesn't seem to show up so much in the cerebellum, but the regional distribution hasn't been studied as well as it should. Good question. Any other questions? Very good. Okay, so if Alzheimer's disease is caused in part by an aneuploidy in chromosome segregation, we asked how we could study this in the laboratory. And of course, the mouse models of Alzheimer's disease gave us an input to that. So what we did was to use in situ hybridization to count chromosome 16, either in neurons or in lymphocytes or in fibroblasts from these animals. For instance, here's one with a presenilin 1 mutation. And you can see that in neurons of these mice, there's about 3 or 4% trisomy 16. If we take a look at another chromosome, it's about the same. So there's a huge amount of aneuploidy in the neurons of these transgenic mice, which are one of the models for Alzheimer's disease. And it's not due to overexpression of presenilin, because if we use a knockout in which the promoter is the same and the amount of expression is exactly what you would expect, but it has that mutation in it that causes human Alzheimer's disease, we see an increase in this trisomy 16. That can also be shown that the reason for that is that the A-beta peptide itself causes chromosome mis-segregation. So in these data, we see, for instance, that trisomy 21 or trisomy 12 is increased by just treating cells with A-beta 40 or 42. And if you count total chromosomes, basically A-beta causes cells to become about 20% aneuploid in 48 hours in culture. That gives us a culprit, shall we say, and now, of course, we'd like to know what the target is. And the target seems to be that A-beta inhibits microtubule motility. This can be shown by just looking at microtubules moving in the microscope upon addition of A-beta. And what we did then was to discover that A-beta actually inhibited certain microtubule motors that are essential for the mitotic spindle to function properly. EG5, KIF4A, and MCAC are 3 of the motors that we found A-beta to inhibit. The normal kinesin heavy chain, for instance, however, is not inhibited by A-beta. Other geneticists have found that these are essential for proper segregation, and if you inhibit them in other ways, you cause chromosome mis-segregation. So this now fits all the data, and we're lucky because some of these motors have been targeted by the pharmaceutical industry before. The reason being that if you inhibit mitosis, you inhibit cancer. Cancer is a big disease, and so it's very nice to make inhibitors of mitosis. And one of these inhibitors is called monostril, and there are other similar ones, which is a targeted inhibitor of the EG5 motor. If you add monostril to cells in culture, you induce a massive increase in chromosome mis-segregation, just as you did with the A-beta peptide, which also inhibits this motor. So that sort of confirms that we have a new target for A-beta, which causes chromosome mis-segregation, and I'll get to in a minute how we might use that fact. Now, if A-beta inhibits this motor in mitosis, the question we had is, could the motor also be present in neurons? And the answer is yes. Here's a staining for EG5 in neurons. You see it in the growth cones and in the cell body. And that means that the neurons have apparently recruited these motors, including the others, for neuronal purposes, in which the cells, of course, usually don't divide. That then begged the question, if A-beta can inhibit the motor in dividing cells, does it inhibit the motor in neurons? And the answer is yes. So here's an example, for instance, that we can detect the EG5 motor activity in the brains of normal mice, but in the APP or the APP-PS mice, there's essentially no detectable EG5 activity. So in vivo, this motor has been inhibited in those cells, and we can show it in the neurons directly as well by just treating neurons with the A-beta peptide. What happens to those neurons is that their microtubules get disaggregated by the addition of either A-beta 1 to 42 or monostril, so that the microtubules don't function properly thanks to the inhibition of this motor. And if you ask what the consequences of that are, and this speaks very strongly to what Bill just told you, it turns out that those motors are used to move proteins and the vesicles in which those proteins are encapsulated from the Golgi out to the cell surface. So if we measure the cell surface levels of, say, the P75 TrkA receptor or the NMDA receptor, they're about half normal in cells that are expressing the APP protein or are treated with monostril, or cells in culture treated with A-beta peptide itself. So that means that not only does the A-beta peptide inhibit this motor and disrupt mitosis, the A-beta inhibits this motor and disrupts normal neuronal function. If you don't have the receptors on the cell surface, trophic factors don't work properly, neurotransmitters don't work properly, and those neurons are essentially working at half speed. This can be shown by inhibiting LTP with A-beta, which has been shown by many people, but now we've shown also that if you treat slices of mouse brain with minostrol, it also inhibits LTP. For those who don't know, LTP is the closest sort of mimic of memory in a slice of brain, where you can detect neurons talking to each other and learning from that talking. No problem. So the conclusion is that A-beta peptide causes mitotic errors. I won't go through the details. It causes neuroplasticity errors, and both of those are important for Alzheimer's disease and Down syndrome. It also suggests that maybe some of the cognitive problems of Down syndrome in their young age may also be due to the inhibition of this motor and the neuronal defects that accrue to that inhibition. So what are we going to do about these new data? Well, there are many potential Down syndrome medicines that have come from Alzheimer's disease research, beta and gamma secretase inhibitors, immunotherapy, potentially inhibition of the ApoE A-beta interaction. We've also shown that caffeine protects the Alzheimer's mice from developing cognitive deficits, and there's good epidemiology in humans. A new one that we discovered from our study of arthritis is now in clinical trials and may be a benefit to Alzheimer's patients, but by the same arguments, it could be beneficial to Down syndrome individuals. And finally, we have an A-beta EG5 interaction that may be worth developing new inhibition molecules for. So do we have the tools? Yes. We have people if we can get them funded, but that's the problem. Getting to people to do the work requires funding. And that means public awareness, political awareness, and foundation awareness of the relationship between Alzheimer's disease and Down syndrome. And the results, of course, will be more self-reliance, more financial security, and less fear for Down syndrome individuals and their families. And I'll stop there, and thank you very much for your attention.
Hello, I'm Ruma Chaudhary from Cambridge Center for Brain Repair. And it's very interesting point that, as you say, the trisomy increase even in Alzheimer. And so do you think these proteins could be age-related? Like if you are young, maybe your odd proteins could be very good clearing process and it will be clear. But maybe Alzheimer's disease or Down syndrome, maybe there is a defect like Ki4 or tau, or could be more sortilin, or could be anything. Could be that is also the problem?
Yes, I completely agree with you. And one of the difficulties of both of these diseases is that they are developmental disorders in the broadest sense of that term. Many diseases that we face in the clinic, somebody gets sick, get an infection or a problem with their diet or something, and they show up with symptoms. You treat the underlying cause and the symptoms go away. The problem with developmental disorders is that the damage accumulates, and you may need to block the initiating event much, much earlier than you can even detect symptoms, which makes things difficult. And that, as Bill said, is what's so important about Down syndrome. We know from the beginning that they're going to develop cognitive deficits and Alzheimer's disease, and therefore we may be able to intervene early.
Thank you for the presentation. I'm Bill Nuttall. I'm just a parent, and I'm from the Hertfordshire local support group up on Downs. You said something that caught my attention, which is that A-beta inhibits mitotic motors. And I'm inferring that might be occurring in every cell of the body, not just the brain tissue. And then you said that pharma has been interested in this inhibition as relevant for cancer inhibition. And anecdotally, I've heard that people with Down syndrome have a surprisingly low level of hard tumor cancer. Just wondering if you had any comment on my anecdotal impression.
It's more than anecdotal, and I find that extremely exciting because I didn't have a chance to go into it, but we've found that the amount of A beta peptide is critical for proper chromosome segregation. Too much causes problems, and too little causes problems. And therefore, if in Down syndrome you have an inhibition of this EG5 motor, then they very likely will have less hard tumors. And that might be a very interesting new mechanism for tumor formation, and may also indicate a potential treatment for the development of solid tumors, in which A beta is actually a medicine by itself.
So my takeaway is this is not just a brain issue.
Absolutely not. Yeah.